So I think it's visible, audible.
Yes, sir.
So a very good evening to everyone over here, right? So this side Dr. Abhijeet, and today yet we are going to discuss the Grand Viva today. So we have initiated this like 45 days plan to MBBS finals, right? So we are revising all the important topics. Hello. Yeah, I think I'm audible now. Yeah.
Yes, sir.
Okay, perfect. Sorry. So, we are going to revise all the important topics in a very crisp, concise, and conceptual manner, right? So, yes, the ones who are going to appear for the practical exam, yeah, yet it's like highly beneficial for the ones who are going to appear. Yes, in majority of the universities, it's like being taught over here right now. So yes, we are going to discuss the Grand Viva under these four headings, right? And I will be discussing five questions from each of these topics today, right? I will be discussing five questions from each of these topics, which I mentioned over here, right? Now, guys, you already know about the Oracle app, right? We have the recorded classes, the live classes, the practical sessions, the exam modules, and we have now the availability of the hard copy book set, right? Which you can order and it will be delivered to your doorstep, right? So yes, the recorded classes, the live classes, the express exam module, the one-to-one doubt clearing session, the practical sessions, right? Even the question bank, guys, considering from the basic to advanced level and containing all the important previous year topics, they are being covered. Now, guys, this is the basic subscription plan which you can see on the screen. And moreover, if you want additional discounts, you can use my code Dr. Abhijeet 20 for extra benefits, right? So the Grand Viva, yes, the first topic that I will be discussing over here today, it is cell enzymes, chemistry and metabolism of carbohydrates and lipids. Now, in that, the very first question, what is an isoenzyme? Can you give an example of one, guys? No. You already know whenever we say isoenzyme, so we are discussing question number one, right? Answer number one. So you already know isoenzyme or commonly called as isozyme, right? It is also called as isozyme. You already know it refers to one of the multiple forms of enzyme. Okay. It refers to one of the multiple forms of an enzyme that catalyzes the same reaction that catalyzes the same reaction. But what is the difference, guys, over here? That it differs. Very important. Should be star marked. You have to tell this, guys. It differs in its amino acid sequence. Amino acid sequence or other properties or any other property or other properties or other properties, right? Despite their structural differences, guys, right? When I say that isoenzyme, it is one of the multiple forms of an enzyme. Despite their structural differences, guys, remember they perform the same chemical reaction. Okay. They often vary in kinetic properties, guys, right? They often vary in kinetic properties. They often vary in regulatory mechanisms or tissue distribution, guys, right? So basically, this is something that you have to remember about the definition of an isoenzyme. But when we say the question says, can you give an example of this? So I will be mentioning like yes, the example, the very common example, guys, it is lactate dehydrogenase. dehydrogenase. And remember this LDH, guys. This LDH exists in several isoenzyme forms. Okay, so they are basically termed as LDH1, 2, 3, 4, and 5. So basically, they are composed of different subunits, guys. You already know they have M and H subunits. M stands for the muscle, and H stands for the heart. Okay, so if for example, guys, if we talk about LDH1, so it has, yes, H4 subunits, right? It has H4 subunits. Now, when I say H4 subunits, so it is predominantly found in which part? That is the heart, right? And red blood cells. Now, when I say LDH5, guys, okay, it is composed of M4 subunits. So basically, it is found in the liver and skeletal muscle. Skeletal muscle. So that is one of the examples that you can remember about this isoenzyme, guys, right? Lactate dehydrogenase, it has like several forms, right? And one you can just see over here. Coming on to the next question, the question says, how does temperature and pH affect the enzyme activity? Now, you guys already know there is something called as optimal temperature. There is something called as optimal temperature. Now, you already know each enzyme, guys. Each enzyme has an optimal temperature range where it functions like most efficiently. Now, temperature is basically this temperature, it is basically close to the physiological temperature of the organism. For human enzymes, it is approximately, guys, you already know it is 37 degrees Celsius, or I can simply say 98.6 degrees Fahrenheit, right? Now, you already know this is the optimal temperature. Now, what happens if there is increasing in temperature or there is increasing temperature, guys? Now, as temperature rises towards the optimal temperature or towards the optimal level, the rate of enzyme catalyzed reaction generally increases, right? So increasing temperature, basically the rate of enzyme catalyzed reaction generally increases, right? Generally increases. Now, why this is so, guys? You already know, because higher temperature provides like more kinetic energy. It provides more kinetic energy to the enzyme, right? It provides more kinetic energy to the enzyme and the substrate molecules. Now, increasing the frequency of the collisions, and thereby the rate of reaction will be further increased, right? So high temperature, again, we have something called as high temperature. Now, when I say high temperature, guys, you should remember that if the temperature exceeds the optimal range, right? Now, what will happen? The enzyme becomes denatured. Enzyme becomes denatured, guys, right? Denatured or denaturation involves the loss of enzyme activity, right? Loss of enzyme's three-dimensional structure, which is like crucial for the activity of the enzyme. Now, if there is a loss of enzyme, I mean denatured of enzyme, that means it is a decrease or complete loss of enzyme activity. Now, guys, coming on to the next one, that is the, yes, again, over here, you can also remember if you have low temperature. If the temperatures are below the optimal range, enzyme activity decreases, rise, okay, because the kinetic energy of molecules is decreased, reduced, fewer collisions will take place, okay, between the enzyme and the substrate. And yes, however, just one point to remember over here, better, that low temperature never causes denaturation, right? Activity can be restored whenever the temperature will rise, right? So yes, that is about how does the temperature like affect the enzyme activity. Coming on to the pH, guys, pH you already know, optimal pH. Optimal pH. Now, intestinal enzymes. If we talk about intestinal enzymes, you guys already know they worked at an optimal pH around 7 to 8, right? If we talk about pepsin, guys, pepsin you already know it's a digestive enzyme and it is found in the stomach. So what is the optimal pH of pepsin? It will be working around 2, acidic, right? So optimal pH is basically the pH level at which the enzyme structure and active site are most favorable, right? So for catalysis, that is something you have to remember. If there is any deviation from the optimal pH, so yes, if there is any deviation from optimal pH, now what will happen? It can affect the ionization. It can affect the ionization of amino acid chain in enzyme's active site and throughout the structure. So it can result in alteration of the enzyme structure, okay, and even the shape of the enzyme and even the charge properties. So basically, yes. So what will happen? Extreme pH will actually denature the enzyme. It can lead to the denaturation of the enzyme, right? Low pH, guys, remember, if we talk about the low pH acidic conditions, right? Low pH means acidic conditions. So acidic conditions can lead to protonation of amino acids. It can lead to protonation of amino acids, and it can also disrupt the enzyme structure and function. Even the high pH, guys. So high pH will be causing deprotonation of amino acid, and obviously it can also disrupt the enzyme activity and function, right? So in summary, guys, you just have to remember both temperature and pH, right, are critical to enzyme activity. So enzyme has a specific range for these factors, right, through which they operate optimally, right? Next question is, how is glucose metabolized in the liver, in the body? Describe glycolysis briefly, guys. Now, glycolysis is the initial stage of the glucose metabolism, and you already know. Now, from the stomach, guys, from the stomach, you already know we have the disaccharide that we are consuming. We have the disaccharides that we are consuming. And obviously, after disaccharides are broken down, we have like glucose, maltose, lactose, but majorly we have like glucose over here. Now, this glucose will be traveling into the liver, right? It will be going into the liver through the portal venous system, right? So, glucose entering into the liver, it can convert into glycogen, guys. It can convert into glycogen, glycogenolysis. Whenever you have like taken food in the well-fed state, this glucose can again go into the cell. Okay, it can go into the cell. Now, in the cell, guys, you already know this is the cell membrane. Now, what will happen? This glucose will try to enter into the cell through GLUT transporters, right? Now we have the cytoplasm over here, cytoplasm. Now what will happen? This glucose will convert into pyruvate, and that is something called as the aerobic glycolysis. Over here, basically the energy will be generated, right? Again, if there is like absence of oxygen, yes, it will be converted into lactate, anaerobic glycolysis, right? So yes, after pyruvate, basically this pyruvate will be entering into the mitochondria, guys. And this pyruvate will be changing into acetyl coenzyme A that will enter into TCA cycle and that will provide energy, right? So this is how the metabolism like takes place. And this glycolysis is nothing but, guys, you already know it is from glucose to pyruvate. You already know from glucose what is formed. Yes. Okay, let us just write down. If in short, guys, you want to know, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, right? They again deviate into dihydroxyacetone phosphate, right? Glyceraldehyde 3-phosphate. Again, all the series of reaction, and ultimately they will be forming pyruvate. Yeah, so that is around this. Now, so that is, you have to remember how the glucose, since it's 1 mark, we just have to like mention all the, and we have already discussed glycolysis in details, right? So yes. Now coming on to the next question, describe the role of essential fatty acids, right, in the body. Now, you already know the very first one that you have to remember, they are responsible for the cell membrane integrity. Cell membrane integrity. Now you already know the essential fatty acids are integral components of the cell membrane, right? They help to maintain the membrane fluidity, very, very important word, membrane fluidity, right? By the cholesterol and yes, obviously the flexibility, right? So this is important for the membrane bound proteins and receptors, right? Cell signaling and all those things, right? So these are the structural components of the cell membrane. Secondly, they are responsible for the production of eicosanoids, right? So essential fatty acids are like precursors of eicosanoids, right? Which are signaling molecules, right? So basically, they include prostaglandins. Yes, the eicosanoids basically include prostaglandins, thromboxanes, right? We have leukotrienes, right? So basically, these are signaling molecules which includes prostaglandin, yes, thromboxanes, leukotrienes, right? So these molecules are involved in wide range of physiological processes you already know, inflammation, blood clotting, right? Immune responses, right? So balance of eicosanoids, you already know they are basically omega-3 and omega-6 fatty acids. Okay. So yes, they are having anti-inflammatory properties. So that is something you can mention. Even apart from that, you can also mention regarding the cardiovascular health, guys. The cardiovascular health. So omega-3 fatty acids, right? So eicosapentaenoic acid or hexanoic acid, you already know omega-6, right? So they have like a positive effect on the cardiovascular system. They basically help in reducing the triglyceride levels in the body, right? So obviously they will also help in lowering of the blood pressure. Okay, they reduce blood clotting. So these are all the roles of the essential fatty acid, reduce blood clotting. Okay, so that is something you have to remember. Okay, they have like neurological and cognitive function as well. So you can just mention about this, right? Now coming on to the next question. Explain competitive and non-competitive inhibition with examples, right? Now in competitive inhibition, guys, you already know an inhibitor competes with the substrate for binding to active site of enzyme, right? Inhibitor basically resembles the substrate, guys, and can bind to the active site, blocking the substrate from attaching. So that is competitive inhibition, right? Now, if we talk about, on the other hand, the non-competitive one, basically it is an inhibitor binds to a site, binds to a site other than the active site of an enzyme, active site of the enzyme. Okay, and that is called as the elastic site. We have already discussed this in detail, elastic site, right? So basically this binding changes the enzyme shape, function, okay, making the active site less effective, inaccessible to the substrate. So that is something you have to remember over here, right? Over here, just examples if you want to remember. Over here in competitive inhibition, remember we have methotrexate, and dihydrofolate reductase. Now, guys, what is this? What is this? You already know methotrexate is a competitive inhibitor of the enzyme dihydrofolate reductase, which is involved in the synthesis of nucleic acid. It is involved in the synthesis of nucleic acid. Now, methotrexate mimics the substrate, that is the folic acid, and binds to the enzyme active site, thereby inhibiting the enzyme, and it interferes with the DNA synthesis. Right now, guys, this mechanism is basically used in the chemotherapy, right? To rapidly or to target those rapidly dividing cancer cells. And that is something this methotrexate is used in the cancer therapy, the chemotherapy, right? Coming on to the non-competitive inhibition, guys, you have to remember, right? We have the examples as the heavy metals, and enzyme inhibition. Enzyme inhibition, right? For example, if we talk about heavy metals, any metal such as lead or I can say mercury, they basically act as non-competitive inhibitors by binding to sites other than the active site. Okay, on various enzymes. For example, mercury, that the example which I have taken, can inhibit the enzyme carbonic anhydrase. Carbonic anhydrase by binding to cysteine residues. Okay, altering the enzyme's conformation and decreasing the activity. So that is again the difference between the competitive and the non-competitive inhibition. Competitive and the non-competitive inhibition. Coming on to the next topic, guys, right? The biological oxidation, vitamin, mineral, nutrition, right? And free radicals, antioxidants, and xenobiotics. Let us just see what all questions are there. Now the question says, what is yes, the biological oxidation, guys, the biological oxidation, and where does it primarily occur in the cell? So basically, you have to remember the biological oxidation refers to the process by which cells extract energy from organic molecules, and through which reactions? Through oxidation reactions. Right? So basically, these oxidation reactions basically involve the transfer of electrons, okay, from one substance to electron acceptor. And which is the electron acceptor, guys? It is oxygen, okay, which helps convert chemical energy into an energy that a cell can use. Into an energy that cell can use, right? Okay. So you already know primary locations. Basically, it primarily occurs in the cell. In which part of the cell, guys? It is the mitochondria. Guys. It is the mitochondria. Right? So mitochondria is the primary site of the biological oxidation. Right? So yes, in the inner mitochondrial membrane, right? Especially in the inner mitochondrial membrane, guys. Mitochondria is a double membrane structure, right? So especially in the inner mitochondrial membrane, you can just remember over here, right? Now coming on to the next one, guys. Explain the role of vitamin B12 and folate. Okay. Yes, in DNA synthesis. Now, very first one, guys, that you have to remember over here, it is the methylation reactions. Now, vitamin B12 is essential for all the methylation reactions that are occurring, right? Basically, it acts as a cofactor. For enzyme methionine synthase. Okay. And you already know what is the function of this methionine synthase. It's involved in the conversion of homocysteine to methionine, right? So this reaction also involves the conversion of 5-methyl tetrahydrofolate to tetrahydrofolate. Basically, it requires vitamin B12. 5-methyl tetrahydrofolate converting into tetrahydrofolate also requires a supplementation of vitamin B12. It also helps in the synthesis of DNA precursors. Now, guys, you already know it is methionine. It is also used to produce S-adenosyl methionine. And you already know commonly it is called as SAM, right? And basically it is a methyl donor in various methylation reactions, including one which is involved in DNA synthesis and repair, right? right? Even vitamin B12, right? It is basically also involved, guys, in maintenance of the myelin sheath, right? Maintaining the health of the myelin. You already know, basically, if we have the nerves, guys, the myelin sheath is covering the axons, right? So basically, this no indirectly supports DNA synthesis by ensuring the overall health of the cells as well, right? Apart from that, if we talk about folate, guys, if we talk about the folate part, now this folate, guys, remember it directly involved in the synthesis of nucleotides, right? So they are building blocks of DNA, right? So yes, folate is crucial for the synthesis of purines, right? Adenine and guanine. We have already told all girls are pure, right? And conversion of deoxyuridine monophosphate, DUMP, to deoxythymidine monophosphate, right? DTMP, right? Which is an essential step in the DNA synthesis. So this folate is basically used for the conversion of deoxyuridine monophosphate to deoxythymidine monophosphate, and basically folate is used, and which is an essential step in DNA synthesis. Even the folate is also required for cell division, guys, cell division, right? So synthesis of nucleic acid and division of cells. So adequate folate levels will ensure that the cells can replicate their own DNA. Okay. And even like formation of tetrahydrofolate, right? So basically it is involved in the transfer of one carbon unit necessary for the synthesis of DNA, right? Coming on to the next one, guys. The question says, what is the importance of iron in the body? What is the importance of iron in the body and how it is involved in the oxygen transport? How it is involved in the oxygen transport. Now, firstly, you should know there is something called as, okay, if we talk about oxygen transport, you already know we have hemoglobin function. Now, iron is a central component of hemoglobin. We have already seen that. Okay, the protein in red blood cells basically responsible for transporting oxygen from the lungs to the tissues. So you already know each hemoglobin is basically containing four iron atoms, which binds to oxygen molecules, right? Even iron is also a component of myoglobin, guys. Myoglobin also, right? So it is a protein, guys. You already know myoglobin is a protein found in muscles. Muscles, basically, it stores and release oxygen as needed, right? Now, myoglobin also facilitates the transfer of oxygen from the blood to the muscle cells. So, especially during the physical activity, guys. So that is something you have to remember. Even the second most important function, you already know the first function is oxygen transport. The second importance you can remember, it is also involved in energy production. And you already know it is the electron transport chain. So basically, iron is in part of various enzymes, right? Iron is a part of various enzymes, various cofactors, right? So basically, it is essential for the function of the electron transport chain in mitochondria, which helps in the production of ATP, right? And ATP is the energy currency of the cell. Then we have the enzyme function. Okay, enzyme function. So metabolic reactions, right? It's involved in metabolic processes, including the synthesis of neurotransmitters, right? The collagen, everything. Even the immune function also. Now, guys, this is something like very, very easy. Explain the concept of a balanced diet, what components it should include, guys. So you already know. Okay. Again, I have not discussed about how it is involved in oxygen transport. So, guys, there are various reactions. You should know it is hemoglobin and oxygen binding, right? Then you already know we have oxygen release, regulation of oxygen affinity, and ultimately we have myoglobin and oxygen storage. right? So there is iron hemostasis that is regulated, guys, right? So iron is absorbed in the duodenum of the small intestine. The body regulates iron levels through various mechanisms such as absorption of dietary iron, recycling of iron from the old RBCs, right? So excess of iron stored in the liver, spleen, bone marrow, okay, it's like bound to ferritin, hemosiderin, and all that. Next question says the concept of a balanced diet, guys. You already know the first thing you should know, there should be adequate energy and energy intake, right? There should be like balanced diet, including all the foods like which provides vitamins, minerals, right? Proportional macronutrients, you already know, like the carbohydrates, right? The carbohydrates, okay? Like it should make around 45 to 65% of the total diet, right? The total daily requirement, right? Apart from that, you should remember the proteins, okay? They should include 10 to 35%. Fats approximately should include 20 to 35%, right? And there should be even the micronutrient sufficiency also. Micronutrient sufficiency also. So basically, vitamin A, C, D, E, B, right? A, C, D, E, and B. And minerals like you already know, we have iron, calcium, magnesium, potassium, zinc. All these irons and like vitamins, the micronutrients, they also should be like taken. And lastly, we have the hydration, right? The hydration, the water intake. What components it should include, guys? You already know it should be like protein sources should be there, whole grains, fruits, vegetables, right? Dietary, dairy foods, and everything, right? Now, coming on to the next question, what are antioxidants, right? And how do they protect the body from free radical damage? Now, guys, free radicals are basically molecules with one or more unpaired electrons. Molecules with one or more unpaired electrons in their outer shell, okay, making them highly reactive and unstable. So these are highly reactive and unstable, right? That is something you should know from free radical. Now, what are these antioxidants? Antioxidants are substances that will neutralize these free radicals, that neutralizes these free radicals, guys, right? By donating electrons to them without becoming unstable, right? So that is, yeah, you have to remember, okay? So yes, how they protect the body, guys? How do they protect the body, guys? Firstly, you already know they neutralize the free radicals. Okay, they prevent cellular damage. They prevent cellular damage, and that is again by neutralizing these free radicals. They protect such structures like the cell membranes, the proteins, DNA from oxidative damage, right? Even they are supporting the cellular repair, right? DNA repair, right? So decreasing the risk of mutations and everything. Examples of antioxidants, guys, you already know vitamin C, vitamin A, beta carotene, that is the vitamin A, selenium, even the flavonoids and everything. So that is you have to remember. Coming on to the third topic, guys, that is chemistry and metabolism of proteins, plasma proteins, immunology, heme metabolism, principles of chromatography, and electrophoresis. Let us see what all questions are there over here. How are plasma proteins synthesized and regulated in our body? Now, you already know the liver, it is the primary site or the main site for the plasma proteins, right? So they produce proteins and it is basically releasing into the bloodstream. So basically, you already know the plasma proteins are albumin, albumin. Okay. Then you already know the main function of albumin. It is oncotic pressure maintenance, the pressure that keeps the fluid within the blood vessels, right? And the second one, it is globulins. Okay, you already know alpha, beta, gamma globulins are there, right? So you already know the alpha and beta, basically they are synthesized in liver. Okay. And gamma globulins are something which are produced by the plasma cells, right? So basically the globulins are involved in transfer of like iron, right? So yes, so even the globulins play a very important role in the immune system. Then the clotting factors, right? Acute phase proteins. So these are all these plasma proteins, right? So acute phase proteins, basically they increase in response to inflammation, guys. I've already told you. Regulation is basically by hormones. And remember, guys, by insulin and cortisol, right? So basically they can like affect the synthesis of this plasma proteins. Now, this if we talk about, for example, cortisol, guys, it can stimulate the production of certain acute phase proteins, right? The second, guys, there is regulation by liver function. Regulation by liver function. Okay, so even the liver health is like responsible, or guys, there can be feedback mechanisms as well, right? So, and even the feedback mechanisms, basically the levels of these plasma proteins are adjusted to maintain the hemostasis, right? So that is how they are regulated, guys. Now, guys, you already know what is jaundice and how it is related to heme metabolism, guys. You already know. Now, jaundice is basically a medical condition, basically caused by the yellowing of skin. You already know yellowing of skin, okay, especially the sclera part of the eye, right? The white part of eye, right? We have mucous membranes, okay? Even the mucous membranes due to elevated levels of or increased levels of bilirubin in blood, in blood, right? Now, bilirubin is basically a yellowish pigment, guys, produced during the breakdown of heme. Okay, it is produced during the breakdown of heme. So basically, you already know heme is a component of hemoglobin, and that is something you have to remember about jaundice. So yes, how it is related to heme metabolism, guys. Remember, firstly, there is breakdown of hemoglobin. Breakdown of hemoglobin. You already know RBCs have a lifespan of 120 days, right? So basically, it is broken down into heme and globin after that. And globin proteins are further broken down into amino acids, which are reused or metabolized. Now, this heme will be converted to bilirubin, guys, right? So, heme oxygenase activity. So, this heme basically undergoes oxidative cleavage by enzyme heme oxygenase, and it will be converted into biliverdin. Again, biliverdin is a green pigment, and then biliverdin will be converted to bilirubin. And finally, there will be transport and metabolism of bilirubin. Okay, we have like unconjugation bilirubin, right? Conjugation will take place in the liver, and ultimately there will be excretion, guys, right? So that is something you have to remember. Now, coming on to the next part, guys, explain the basic principles of electrophoresis and how it separates the molecules, guys. Remember, yes, some like first you should know there is electric field. Okay, so basically this electrophoresis works applying an electric field, right, across a medium. Basically, it's usually a gel or liquid, guys, in which the sample is placed. Now, the electric field is created by applying a voltage. It is a okay. Basically, there are two electrodes, and the difference along the two electrodes. Charged molecules, guys, you already know will move towards the electrode with the opposite charge. Anions, basically negative charge, move towards the positive charge, that is the anode, while positively charged will move towards the negative charge, that is cathode. Again, guys, you have to remember about the medium, guys. Medium, that is the gel electrophoresis. Gel electrophoresis, and we have the buffer solution. Buffer solution. Gel electrophoresis and buffer solution, right? So the most common medium basically in the gel electrophoresis, it's basically an agarose or a polyacrylamide gel, right? So yes, after that there is separation of mechanism, or like there is separation mechanism, right? So they are basically separated on the basis of size and charge, right? So the molecules separated based on their size and charge, right? You already know the ones who are having like smaller molecules, like they move faster, travel through gel matrix, and the larger ones, than the larger ones, guys, right? And finally, we have the final stage called as the detection or the staining stage. Staining stage, right? Now, what are essential amino acids, guys? What are essential amino acids? And you already know, yeah, the essential amino acids are the ones which are not synthesized our body. We actually take it from outside. That's why they are essential, and body cannot produce them, right? So basically, the amino acids, um, they are not synthesized by the body, not synthesized by the body, right? And they are crucial for like they are taken or ingested through outside, right? So yes, right? Why you already know what are essential amino acids, right? So we have histidine, isoleucine, leucine, lysine, methionine, right? All these are essential. Like we have 8 in number, right? We have phenylalanine, threonine, tryptophan, valine, right? So that is something you have to remember. And why they should be obtained from the diet, guys, I have already told you they are not synthesized. There is lack of biosynthesis in our body, lack of biosynthesis, right? We have like health implications, health implications, right? Protein malnutrition and all that. So that is something you have to remember. What are the main components of the immune system, guys? So firstly, you should know we have the cells of the immune system, that is WBCs, that is the leukocytes, right? Leukocytes, you already know we have neutrophils, we have lymphocytes, okay? Lymphocytes, that is the B lymphocyte, T lymphocytes, right? B lymphocytes, T lymphocytes are there. Even guys, you should remember, yeah, we have helper T cells, right? In T lymphocytes, we have helper T cells, cytotoxic T cells, right? Regulatory T cells, monocytes, macrophages. Again, there are many other as well. Monocytes, macrophages. Okay. And yes, that is the main components of the immune system. We have primary and secondary immune system, right? The primary lymphoid organs, you should also know the bone marrow and thymus. And secondary lymphoid organs, you already know spleen, tonsils, appendix, Peyer's patches, lymph nodes, right? So that is the main components of the immune system. Coming on to the next topic, guys, start the chemistry and metabolism of nucleotides, molecular biology, oncogenes, tumor markers, and organ function test. Now, the very first question that they have asked, what are the liver function tests, right? What are the liver function tests? So basically, they are the group of blood used to assess the health and functions of the liver. And basically, they measure various proteins, enzymes, right? So that can detect the liver damage. And the ones which you have to remember, we already know we have alanine aminotransferase, right? We have aspartate aminotransferase, AST, alkaline phosphatase, right? We have gamma glutamyl transferase, right? So that is you have to remember. Even the ones like these are measuring the enzyme the liver enzyme levels. If we talk about the protein levels, guys, protein levels, you already know we have the serum albumin levels, right? Low level can indicate again the liver disease. The total protein levels, the total protein, bilirubin, right? And we have like other tests also, guys. You already know we have prothrombin time. Okay, INR, that is international normalized ratio. So basically it measures the blood clotting time derived from prothrombin. It is used to monitor patients on anticoagulant therapy. So elevated INR can actually indicate liver dysfunction and problem with the coagulation process. So that is something you have to remember over here. Okay. Coming on to the next one, function. Okay. Yes. Next question, what is the role of P53 gene? Guys, you already know it is tumor suppression gene. It is the tumor suppression gene, right? The second one, it is apoptosis or the programmed cell death, right? Then we have senescence, that is aging, right? And regulation of metabolism, okay? Role of RNA polymerase in transcription, guys. RNA polymerase in transcription. So you already know we have initiation, okay? And okay, you already know about the initiation. So in transcription, the very first process, or it is initiation, guys. Transcription begins when RNA polymerase will bind to specific DNA. It binds to specific DNA, guys, and DNA sequence that is called as promoter, which is the upstream of the gene. We have already discussed this, guys, right? So yes, then it is recognition. It also helps in recognition, guys. The RNA polymerase is guided to the promoter by various transcription factors. So in prokaryotes, RNA polymerase directly binds to the promoter region, right? So that is something you have to remember. Even they are also helping in the formation of transcription bubble. Formation of transcription bubble. RNA polymerase unwinds the DNA double helix to create a small localized area. Okay, that is called as a transcription bubble. Now describe the role of nucleotides and DNA and RNA synthesis. Now, you already know the nucleotides. I even don't need to tell you, guys. We have the nitrogenous base, a pentose sugar, and yes, the phosphate group. So that is the composition of the nucleotides. That is something you guys already know. And yes, we have the nucleotide components, guys. You already know that nucleotides used in DNA synthesis are deoxy, okay, deoxyadenosine triphosphate or deoxycytidine or triphosphate, right? So all these that are used in like nucleotide components in the DNA synthesis. Okay, then DNA polymerization is there. Okay. After that, guys, is something like very, very easy that you already know. Even yeah. So the last question, guys, how is glucose tolerance tested? And yes, and what does it indicate about the pancreatic function, guys? You already know, right? We have something called as oral glucose tolerance test, right? So glucose tolerance test, basically it is used to assess how well the body process glucose, and it evaluates the pancreatic function, guys, right? And how the pancreas, like they're specifically the pancreas able to produce the insulin or not. So basically they are testing, and we have the oral glucose tolerance test, right? So yes, obviously we have preparation, interpretation, guys. You already know fasting blood glucose levels. If it is more than equal to 126, okay, then you already know if it is more, it is like diabetes is confirmed. 2-hour blood glucose testing. If it's more than 200, then again it is diabetes. It is confirmed. Now, yes, we have intravenous glucose tolerance test also, right? So yes, we can do that as well. That was about today's class, guys. So these are all the important questions that we have already discussed in our regular classes, but in 1 mark, we just have to like briefly explain about that. So thank you for watching, and here's the like you can just go through all these links, right? Thank you.